Alright, so this week's lab we're going to be looking at what's called signal transduction. And we got these 36 trillion cells in our body, and so there's got to be some modes of communication. And that essentially is how this is taking place. This communication takes place through this signal transduction that will be taking place between the cells. Alright, so there's going to be a couple ways in which this communication can take place. We can take communication by the cells by direct connection between the cells or through chemical signal. We're going to spend most of our time here, but let me just show you the difference between the two. In the communication between two cells, there is a time when some cells have what's called gap junctions. Okay, we're going to see this when we talk the cardiac cells, the cardiac muscle cells, there are gap junctions. And in these gap junctions, these are proteins, these connexin proteins, that actually make a tunnel between cell 1 and cell 2. So the cytoplasms from each cells have access to each other through these gap junctions. So molecules can move through here, ions, small molecules, and so we can get communication across to the different cells via these gap junctions. Okay. The one we're going to be spending most of the time today is chemical signaling. And you can think of this as how we communicate. We have, again, somebody has to talk, use their mouth, words come out, and then we have the ears to listen. You can think of the cell here that's going to release a chemical as the talker or the mouth, and it releases the words that are the chemical messenger. And for a cell to receive that message, it has to have a receptor. Okay, so we need those three components. We need a cell that releases a chemical, but the cell that is responding or listening to the signal has to have a receptor. That is the way the cell listens to what's going on. Okay, so again, we're going to spend most of our time looking at this difference of chemical messaging. All right, so we're looking at chemical messaging, the signal transduction and chemical messaging. So let's first look at kind of the class of chemical messengers. One class is what we call paracrine messengers. Okay, this is going to be where cells can talk to the cells locally, next to them, very close to them. All right, you have one cell, the secretory cell. Okay, that is the cell that's going to release the chemical. In this case, the chemical is a paracrine messenger because basically it is releasing the chemical into the interstitial fluid around it. Okay, therefore it can only talk basically to cells that are nearby that have access to this interstitial fluid. Okay, so again, this is localized communication. Okay, our lab today, we're going to be looking at one of the signals we're looking at is a paracrine messenger. Okay, but again, the only cells, if there's a cell over here that doesn't have a receptor, it can't listen. It can't respond to this chemical messenger. Only the cells that have a receptor are going to respond to that chemical messenger, in this case, a paracrine messenger. Okay. Some modes of long-distance communication. First one class is neurotransmitters. Okay. Neurotransmitters are released by neurons, and this communication can take place over long distances. This cell body here of this neuron could be up in the spinal cord, and where the chemicals release at the axon terminal, this could be all the way down at your big toe. So this whole axon could be several feet in length. So you can see we can get long distance signaling. Okay, the neurons are one of the main communication pathways to be able to communicate and coordinate cells, and they're able to do this over long distances. Okay, but the chemicals that the neuron releases are neurotransmitter. But the concept is the same. The neuron releases the neurotransmitter, and basically releasing in the small little synapse where the target cells are going to have, again, they have to have receptors to respond. Okay, so again, we got one cell releasing chemical messenger, and the other cell has to have the receptor to respond to that chemical messenger. Okay? In this case, for neurotransmitter, can take place over long distances, but it is the chemical messenger is being released by the neurotransmitter, or sorry, by the neuron. Okay. Another form of long distance signaling can take place via our endocrine cells. Our endocrine cells secrete chemical messengers called hormones. All right, and where do the endocrine cells, secretory cells, secrete their hormones is into the bloodstream, okay, into the blood vessels. So therefore, any cell that has access to the blood supply essentially has access to the chemical messenger, the hormone. Okay, but again, what cells respond to the chemical messenger? Okay, non-target cells, cells that won't respond, don't have a receptor. Okay, but the cells that are going to respond to that chemical or the hormone in this case are the cells that have receptors. Okay, we've looked at our insulin signaling and our homeostasis. Many cells don't have receptors for insulin, but the target cells are your liver cells and your skeletal muscles. They must have the receptor to respond to that insulin signal. Okay, so that's for the one side of the equation, in that we have the different chemicals. Now we're going to look at what happens when these chemical messengers bind to the various receptors. So we're going to look at the different class of receptors for these chemical messengers. So the first class of receptor we're going to look at here is ligand-gated ion channels, also called ionotropic receptors. Okay, but you can imagine what they are. They are essentially ion channels. Okay, we've already seen ion channels with our membrane transport, but these are those gated channels I briefly mentioned, in that what's going to open them is the binding of a ligand, or in this case, ligand being the chemical messenger that is released. Okay, you can see here is an ion channel, okay, a ligand-gated receptor. It is closed. There are no ions moving through. When the chemical messenger or the ligand binds to the receptor, now the channel is open, and now ions, depending on what kind of channel, what kind of receptor is, depends on what ions can move through. Some channels allow multiple ions, some only allow single individual ions. Okay, so they're usually selective on what ions move through here. Okay, the simplest form is if we have sodium, potassium, or chloride ions can move through. Essentially what that's going to do is change the membrane potential of the cell. Okay, we can also have ion channels that allow calcium, and you can see we can get much broader effect with the influx of calcium. We can change electrical properties similar to what we see here, but you can see we can activate what we call a second messenger system, and this calmodulin. We can cause muscle contraction or also secretion of other molecules. Okay, but this process is the same. Basically, binding to that chemical messenger opens a channel to allow the various ions in. Okay. Another class of receptor find is we got enzyme-linked receptors. Okay, and this essentially, basically this receptor has an enzyme on the intracellular portion of it. All right, without the chemical messenger binding to the receptor, that enzyme is inactive. It's not going to catalyze the reaction. Okay. Binding of the chemical messenger, though, causes a conformational change in the receptor, and that activates the receptor. A common type of this enzyme-like linked receptor, which we're going to find, is a tyrosine kinase. So just what's in that name, knowing what enzymes are, kinase is going to add a phosphate group, and in this case it's going to add it to a tyrosine amino acid in a protein. Okay, so you can see now active. Here's the protein that has a tyrosine residue in it, and we're going to add a phosphate. That is what this enzyme catalyzes, is adding that phosphate group to that tyrosine of that protein. Okay, that can activate other proteins, and we can get a response in the cell. Okay, a common type of tyrosine receptor or tyrosine kinase receptor is that of insulin. Insulin binds, and then we get an activity where we get glucose channels brought to the membrane. Okay, our lab today we deal with VEGF. The VEGF receptor is a tyrosine kinase receptor. So binding of that VEGF will cause activation of the enzyme, and we'll get phosphorylation of proteins that will cause activities in the cell. So the next class of receptor is kind of the most prevalent of receptors you're going to find out there, and there are multiple responses that can take place while using what we call a G-protein couple receptor. Okay, G-protein couple receptors essentially is that. You'll see here is the receptor. Okay, and it is coupled with these G-proteins coupled with it on the intracellular side. Okay, these G proteins are inactive when the chemical messenger is not bound. Okay, once the chemical messenger is bound, that activates the G proteins, and now the G proteins, there we'll see, can have a wide range of effects. One of the effects are it can open up ion channels. Okay, so here, chemical messenger binds, G protein gets activated, uncouples, and goes and opens ion channels. Okay, kind of similar to what we saw with the ligand-gated. This is a slower response than saying those ligand-gated ion channels, which basically when it binds, boom, the channel's open. We got to go through these processes to get this ion channel open. What we're going to see is the G proteins, besides opening ion channels, we can activate what's called second messenger systems in the cell. Okay, so let's take a look at one second messenger system. What we're going to see is these second messenger systems can basically amplify the signal from basically one messenger. One of these systems is the phosphinocidal system, in which activation of the G proteins will go and activate this phospholipase C enzyme. Okay, and we'll see this can activate IP3 and diacylglycerol, DAG, but you can see what we're getting is we're getting these activations of these cell signalings. This is more of a cell molecular class, but what you can see is this response activating in the phospholipase Cs, we can get multiple responses through the cell, depending on what cell it is and what receptor is being activating this phospholipase C. Okay. The second messenger system I kind of want to take a closer look at is one of the more common is a cyclic AMP second messenger system. Okay, and this gets activated by, again, a G protein couple receptor. All right, we're going to walk through this so we can take a look. So I want you to this gives you a good idea of what's happening with these second messenger systems. Again, binding of the messenger activates the G protein. The G protein goes, in this case, in this system, it activates adenylate cyclase. Okay, this is an enzyme that catalyzes, takes ATP, and converts it into cyclic AMP. Okay, now cyclic AMP can go and activate proteins, and those protein kinases can then phosphorylate more proteins and get activation of these proteins, and we get a response in the cell. What we can see is by this binding of this one messenger, we're going to see we can get millions of proteins activated in the cell. So we're getting this through this pathway here, we're getting an activation inside the cell. So binding of one messenger gives us this widespread effect in the cell. Okay. So let's take a Alright, so let's look how this amplification takes place with our cyclic AMP system here. Here's our receptor. We get one chemical messenger binds to one receptor, and binding here can actually activate multiple G proteins. There can be multiple G proteins coupled here, and say we activate 10 G proteins by binding to one receptor. That allows those G proteins to each go activate adenylate cyclase enzyme, right? And those 10 adenylate cyclase can catalyze many reactions, producing, let's just say, 5,000 cyclic AMP molecules. So now we have 5,000 cyclic AMPs that can go down and activate 5,000. Each of those cyclic AMPs can activate a protein kinase. Right? And now this enzyme, it's a kinase, it's an enzyme, can in turn phosphorylate, say each one can phosphorylate, say 5,000 proteins, and we get activation of millions of proteins. So you can see how binding of one chemical messenger, we can get to millions of proteins activated in the cell. So we get this amplification of the signal. So all those chemical messengers so far, or those receptors we've looked at, what have you seen? They've all been here on the plasma membrane. Okay. So what kind of chemical messenger are we thinking about polarity-wise? Okay, larger polar molecules, can they make it through the plasma membrane? No, they cannot. So they can act, but they can act on these receptors that are on the cell membrane. Okay, but there are some chemical messengers. Classic examples are your steroids, your progesterone, your estrogen, your testosterone. Those hormones are lipophilic. Okay, they are nonpolar. They are basically a derivative of cholesterol, which is a very small nonpolar molecule. So these chemical messengers, they don't have to act on a membrane receptor. Okay, their receptors can be intracellular. Okay. So a lymphophilic messenger, they can have, here's your receptor. It can be either cytoplasmic. Okay, the receptor can be sitting in the cytoplasm, or you can have a nuclear receptor sitting in the nucleus, because these lipophilic messengers can make it through these membranes. They can zip right through because they are small and nonpolar. All right. These intracellular receptors have a very distinct effect in that binding of these intracellular receptors they have a domain. They have a binding domain for the chemical messenger, but they also have a DNA binding domain on this receptor. And so when these receptors are bound and activated, they go and bind to portions of the DNA called a hormone response element. This would be the beginning of a gene. So what the effect of these intracellular receptors are they act as transcription factors because they are going to induce transcription of various genes that have response elements that bind to these hormone complexes. Okay, so but they are not bound, they are not activating and promoting transcription unless the chemical messenger is available. Okay, so ultimately these intracellular receptors result in transcription and ultimately translation of proteins. Okay, so you can see this as a slower effect than maybe what you see at the plasma membrane, but you can see it makes proteins, so it has usually these intracellular receptors will have a longer term effect on the cell. Okay, so that's intracellular. For our lab today, we have to discuss one other aspects. Okay, many of your drugs, many of your medicines, your pharmaceuticals, they act as receptor antagonists or receptor agonists. Okay. Here's your natural ligand. Okay, binding here, activates, gets a response. There are many medicines that are agonists, and basically an agonist is not the actual natural molecule, but we could take a medicine and it has a molecule that will bind and activate a receptor and get a similar response. Okay, think of just from a neuro standpoint. Okay, amphetamines, everybody's heard of amphetamines. Okay, get you wired up. It's because they act as dopamine receptor agonists. Basically, they mimic dopamine and they activate the receptor and they give a response as if there was a lot of dopamine there. Your reward centers act on dopamine, and so therefore you get these effects, and that's why they're addictive too. And on the opposite end, an antagonist would block the receptor. Okay, if this antagonist is here, then the natural ligand cannot bind, and that prevents a response happening at the cells. From a neural standpoint, again, there are dopamine antagonists for like schizophrenic drugs. Schizophrenic have hallucinations. There's a dopamine 2 antagonist that blocks that dopamine 2 receptor, and it will prevent hallucinations and delusions. Okay, because it's blocking that effect. Right? Because in our lab today, you're going to be looking at basically either an agonist or an antagonist and looking at how it might help treat cancer. And the actual, okay, in our lab today, the signaling we're going to be looking at is one that comes from chemical messenger is vascular endothelial growth factor, or VEGF for short, and looking at the signaling that takes place at the VEGF receptor. Okay, binding of the VEGF receptor at the endothelial cells signals them to produce more and basically do what's called angiogenesis, growth of blood vessels. If you need to get more blood supply, you want more blood vessels to that area so you can supply more blood. We're going to look at this in effect to cancer cells. What do cancer cells do? They are constantly dividing. Okay? Usually this division of these cells is well regulated. There is lots of regulations put on the cells to keep them from continually dividing. But cancer cells have got an ability to continually divide. So these constantly dividing cells, you can imagine division takes a lot of resources, a lot of energy for the cells, and so there needs to be a good supply to these cancer cells to be able to grow successfully or divide successfully. Okay, so many times you'll see in cancer cells there is a rich blood supply to them. Okay, so what you're going to be looking at is, can you use a drug to affect the VEGF signaling to maybe result in treating cancer, okay? Limiting the tumor growth.